Picture this, it's 1985. The music world is buzzing with the Sony Walkman. This revolutionary device lets you carry your favorite tunes everywhere. Technology is changing fast. Now imagine someone whispering about an even wilder future. What if we could edit the very building blocks of life? What if we could rewrite our genes like songs on a cassette tape? That future is closer than you think. Welcome to the world of genetic engineering. The journey began long before the Walkman, even before the first computer. It started with a simple observation. Living things can change over generations. Humans have been shaping the traits of plants and animals for millennia. Think of the difference between a wolf and a pug. Both are canines, yet they look and behave so differently. This transformation is the result of selective breeding, a crude form of genetic engineering. But to truly understand how it works, we needed to dive deeper into the heart of life itself. We needed to uncover the secrets of DNA. Imagine a world without supermarkets. Our ancestors relied on farming for survival. They carefully selected seeds from the best plants, hoping to grow bigger fruits or more resilient crops. This ancient practice, repeated over generations, led to the domesticated plants we rely on today. What they didn't know was that they were manipulating the very essence of life, DNA. Fast forward to the mid-20th century. Scientists in lab coats huddled over microscopes in dimly lit rooms. They were locked in a race to decipher the structure of DNA. In 1953, the breakthrough came. James Watson and Francis Crick unveiled the double helix, the elegant spiral staircase that carries our genetic code. This discovery revolutionized biology. We finally had the key to understanding how traits are passed down through generations. But the story doesn't end there. The discovery of DNA was just the beginning. It opened a Pandora's box of possibilities. What if we could go beyond simply selecting desirable traits? What if we could directly edit the genetic code itself? The 1960s marked a turning point. Scientists, emboldened by their newfound knowledge of DNA, ventured into uncharted territory. They began experimenting with mutations, those random changes in the genetic code that can lead to new traits. Some researchers used radiation or chemicals to induce these mutations, hoping to create plants with higher yields or resistance to diseases. In 1972, a landmark experiment took place. Scientists successfully inserted a foreign gene into a bacterium. This groundbreaking feat proved that we could transfer genetic information between different organisms. It wasn't long before researchers applied this technology to animals. In 1974, the first genetically modified mice were born, carrying genes from other species. The era of genetic engineering had truly begun. The 1980s saw the rise of genetic engineering as a commercial force. Biotech companies sprung up, eager to capitalize on this revolutionary technology. Their first target, the food industry. In 1994, the Flavor Seva tomato hit supermarket shelves. This genetically modified tomato, engineered to ripen slower and last longer, marked a turning point in our relationship with food. Since then, genetically modified crops have become increasingly common. From corn-resistant to pest to soybeans, engineered to tolerate herbicides, these crops have reshaped agriculture. Proponents argue that they increase yields, reduce pesticide use, and enhance nutritional value. Critics, however, raise concerns about potential environmental impacts and the control wielded by large corporations. The debate over genetically modified food continues to this day. Section 5. Editing Humanity. The dawn of designer babies. The 1990s ushered in a new era of genetic engineering, the modification of humans. In 1996, Dolly the sheep, the first mammal cloned from an adult cell, was born, sparking ethical debates about the implications of cloning. But cloning was just the tip of the iceberg. Scientists were developing techniques to directly edit the human genome, raising the prospect of designer babies. One of the first breakthroughs in human genetic engineering came in the form of mitochondrial replacement therapy. This technique, pioneered in the 2010s, allows doctors to replace faulty mitochondria in a mother's egg with healthy ones from a donor. This procedure prevents the transmission of serious mitochondrial diseases from mother to child. Although not gene editing in the strictest sense, it represents a significant step towards altering the human germline. Today, genetic engineering is no longer a futuristic fantasy. It's a reality that's rapidly changing our world. CRISPR-Cas9, a revolutionary gene editing tool, has made it faster, cheaper, and more precise to alter DNA. This technology holds immense promise for treating genetic diseases, developing new therapies, and even enhancing human capabilities. Imagine a future where we can cure inherited diseases like cystic fibrosis and Huntington's disease. Imagine a world where we can engineer crops that can withstand the challenges of climate change. Genetic engineering has the potential to solve some of humanity's most pressing problems, but it also raises profound ethical questions. As we delve deeper into the realm of genetic engineering, we must tread carefully. The ability to edit the very essence of life comes with enormous responsibility. Who decides which genes to edit and for what purpose? Will these technologies be accessible to everyone, or will they exacerbate existing inequalities? One of the most pressing ethical concerns is the possibility of unintended consequences. Altering one gene can have ripple effects throughout the genome, leading to unforeseen side effects. Moreover, the long-term impacts of genetic engineering are still unknown. We're sailing in uncharted waters, and it's crucial to proceed with caution and humility. The future of genetic engineering is full of possibilities and uncertainties. Will we one day be able to enhance human intelligence, lifespan, or physical abilities? Will we create new organisms with novel capabilities? The possibilities are both exhilarating and unsettling. Some ethicists argue that we should draw a clear line between therapy and enhancement. Treating genetic diseases is one thing, but using genetic engineering to enhance human capabilities raises concerns about eugenics and the creation of a genetic elite. Others believe that we should embrace the transformative potential of genetic engineering, arguing that it's part of our ongoing evolution as a species. The journey from selective breeding to gene editing has been long and winding. From the humble farmer selecting seeds to the scientist editing DNA with CRISPR, we've come a long way in our quest to understand and manipulate life. Genetic engineering has the power to reshape our world in profound ways, offering both immense promise and daunting challenges. As we stand at the cusp of this genetic revolution, we face a fundamental question. What kind of future do we want to create? The answer lies not only in the hands of scientists and policymakers, but in the hearts and minds of every individual. For the future of life on Earth, in all its diversity and wonder, is ultimately a shared responsibility.